Hello, I'm Joseph Yu from the Department of Computer Science here at Baylor University. Today, I'll be presenting my semester project in data mining. My topic is called Predicting Gene Expression Using Graph-Based Deep Learning Approach. On a high-level overview, my project will be introducing a novel approach for gene expression prediction that is vastly different from the current approaches that favor a linear additive model. My approach uses a graphical representation to represent the interactions within the human genome and uses graph spectral embedding as my model input for gene expression predictions. The motivation for the project is to design a new alternate approach for gene expression prediction from the traditional models. This assumes an additive linear model, which has been shown to work well, but does not fully consider the spatial structure of the genome. In gene expression, the three-dimensional structure heavily influences the transcription, gene transcription, given our current understanding of the human genome and its molecular processes. And another motivation is to provide a working pipeline for converting the three-dimensional structure of the human genome into a structure that can be easily used for modeling. And lastly, we present the visualization of human genome in a graphical representation, which can potentially lead to more research questions on the human genome by viewing the genome as a graph instead of thinking of it as a linear structure. So we started with the background of the project, which is the human genome. Most people are familiar with DNA and can imagine the double helical structure of the human genome with its A, T, C, and Gs. But what a lot of people often overlook is the fact that the human genome is three-dimensional and can form complex twists and loops. The resulting secondary spatial structure of the human genome creates these physical separations within the human genome, effectively sectioning off different parts of the genome like we see in the figure here, where we see a loop structure, which we call a chromatin loop. That forms, which often contains genes and non-gene regions that can contribute to gene expression. And gene expression affects human protein levels within the body and can affect the overall phenotype of the individual. And this is very important because scientists are very interested in the mechanisms and effects of gene expression. A large part of this tied to the tissue-wide association studies, or TWAS. TWAS uses gene expression prediction to help associate gene variation to phenotypes. And in most research topics are often related to hereditary diseases or complex diseases that has huge health implications for humans. For example, many cancer types are heavily influenced by genetics, as well as neurodegenerative diseases, such as early onset Alzheimer's, Parkinson's, etc. And TWAS in gene expression is a popular research method that scientists are using to investigate these diseases. Currently in TWAS, the most popular approach for predicting gene expression is the use of additive linear models, which we mentioned earlier. And this is a simple linear function that assumes different parts of the genome contributes a certain amount to the expression of a particular gene. And by adding up the different weights, the function generates a value that is essentially the predicted expression level of a gene. So many popular models employs this assumption, such as PredictScan and joint tissue imputation. They all use this assumption on predicting gene expressions. However, by approaching the problem from a different perspective, such as using the spatial structure or three-dimensional structure of the human genome, it may be possible for us to create an alternative model for gene expression that works equally well or even better, based off of our understanding of the three-dimensional human genome structure that we talked about earlier. So here we present the experimental pipeline that we designed for this project. We're using data sets from two sources, both of which are publicly available. The first one is the GeneHancer dataset that contains the interactions between genes and non-gene regions of the human genome. and from which we can create an adjacency matrix that represents the genome interactions in a graph. The second one is the gene expression data from the GTEx project, which stands for Genotype-Tissue Expression Project, and contains the expression levels for each human gene in different tissues. Although for our project scope, we are only using the tissue levels from the human liver, but we do plan on expanding this from liver to other tissues as the project develops. So that's the first part of our experimental pipeline, and the second part of the pipeline, we're using the spectral embedding algorithm to generate the graph embedding of the genome interaction graph, which we create graph embeddings with latent dimensions of 2, 4, and 8. We then take the graph embeddings and train a neural network with dense layers and then evaluate the model performance. We will go into more detail on the motivation and mechanism of using graph embeddings in our pipeline. But that was the second part of our pipeline, and this slide here shows the overall experimental pipeline. It's shown fully on the slide. So we created a genome interaction graph for the entire genome from the GeneHancer dataset and from the GTEx dataset, and where we end up with around 10,000 graphs for training data across all chromosomes combined, and around 150 test data from genes in chromosome 21, which we separated from the 22 autosomes, chromosome 1 to 22, we selected chromosome 21 because chromosome 21 is the smallest human chromosome of the genome. So that way, it's easier for us to set aside a test data set, and also it's easier for us to retain the most training data with our current design. So using the GeneHancer dataset, we visualize the human genome in a graphical format, like what's shown here on the slide on the right, where the red vertices are genes and the blue vertices are non-gene regions that have interactions with the genes. One of the very interesting observations here that we have observed across all chromosomes is that for different genes, the graphs have different sizes. As you can see, some of the genes here is made up of 5 or 4 vertices, whereas some other genes have 10, 20, 30 vertices, all connected in a component. So this presents a special challenge for us when we are planning on using these graphs to train a model to predict gene expression prediction, since every training data example may not be the same size. So from this challenge is from the variable graph sizes. And if our training example have different sizes, it cannot be used directly as training input data for a neural network input layer. So on this slide, we show that some loops are bigger and contain more vertices, whereas others are smaller and contain considerably less elements. Here we show on the right are 2 adjacency matrices from chromosome 21, which each contain genes of their own, and show that one gene contains 25 vertices in its graph, and another one contains 5. So this variable size in chromatin loops and its resulting genome interaction graph made it very difficult to directly use as input data to our neural network when these graphs have variable sizes. So to solve this problem arisen from variable sizing graphs, we sought to find a way to create data examples that have the same size and still retain the structured information for each of the graphs. And the solution that we are using is the spectral embedding technique, where from the original adjacency matrix of the graphs, we first calculate the Laplacian matrix, and then we derive from the Laplacian matrix, we derive the eigenvalue maps from the Laplacian matrix. And at this point, we have a choice to make where we can select how much the dimension space or eigenvectors we want to retain from our eigenvalue map. And in our project, we selected 2, 4, and 8 dimensions as our reduced dimension that is consistent across all examples. so that we would have a uniform shape for input data on the neural network inputs. The project uses spectral embedding from the Python scikit-learn library, and feed the data into the layers shown on the right side of the slide, which has 1024 node vertex at the input layer and 512 neurons in the second, and then 120 in the third, and then 32, and then lastly we have this one single output value that would be the predicted gene expression for the gene. And as you can see, we employ several techniques where we use dropout and also batch normalizations at different layers to help us learn the graphical patterns better from the graph embeddings. And here in the next slide, we'll show our performance of the model. So here we show the performance and using dimension 2 and 8 examples and comparisons. The two figures show that the training progress with our layers and in 100 epochs does not show a meaningful learning progress. However, we do see that with more dimensions, the overall loss seems to be reduced generally in 8 dimensions compared to our 2 dimensions on the left. However, both the 8 dimensions and 2 dimension models have very poor training results and poor performance, and we do not see a lot of convergence or learning between the training loss and the validation loss using 100 epochs. And on this slide, we show for our chromosome 21 test data, we show dimensions 2, 4, and 8. The performance is very similar across all of the 3 different types of dimension models. And we see that there is not a lot of change between the mean squared error or the mean absolute error. And we see that from the R-squared metric that all 3 models have very poor performance in their predictions on chromosome 21, training on the other chromosome genes. So this is our results. And given our model performance, the model does not seem to capture this gene expression pattern as well. And even with the increased dimensions, we did not see an increase in performance. There may be some possible reasons for the result, and one of which may be the choice of graph embedding techniques, where spectral embedding may not best highlight the graphical patterns of our interaction graph, or maybe we need longer training time for the model to fully learn from the data. So stemming from discussions and results, experimenting with other graph embedding techniques may generate better model performance than we currently have using spectral embedding. Uses the Laplacian matrix of the original adjacency matrix. Another possible attention or modification we may attempt is to introduce weights to our edges that provides information on the frequency of contact between the gene and non-gene region in the genome. There are, from the GeneHancer dataset, there's also strength of value, which we may include as edge weights in the future. Since in our current model, the model uses unweighted edges, which assumes equal weights of 1 between any two vertices with an edge. And if there's no edges between them, then it's 0. So these are some of the steps that we could try to attempt to further improve the model. But we have reached the end of my presentation. Overall, in summary, we presented a possible approach for gene expression prediction and how we can convert the complex landscape of the genome interactions into a workable data set for a neural network model that we can train on and model after to predict gene expression. And this slide lists the references of my presentation. Thank you for listening to my presentation, and please feel free to reach out to me for any questions and suggestions for my project at joseph_u1@baylor.edu. And have a great day. Thank you.